Bismillahirrahmanirrahim. The three events in the healing process are cellular migration, changes in the extracellular matrix, and then proliferation of the cell. The first among these, cellular migration, will be discussed in this video. Following are the references for this video. The migration of the cells initiate repair. It begins almost immediately after the injury and is driven by the migration and activation of the specific cells and migration and activation of the specific cells at the site of the wound. This process is triggered by both the mechanical disruption and biochemical mediator. The mechanical cues and local mediators are whenever there is injury, there is changes in the tissue tension and cell-to-cell interaction due to mechanical disruption. Then there is migration of the cell into the site of the wound and activation of the local cells. Both of these lead to release of various biochemical mediators from the platelets, basophils, mast cells, resident macrophages, epithelial cells, and mesenchymal cells. The platelet activation is triggered by exposure of the collagen at the site of the endothelial injury. The activation of the platelet initiates hemostasis via aggregation and cross-linking of the fibrin. There is also release of platelet-derived growth factor, cytokines, chemokines, various proteases, vasoactive substances, and all these promote the formation of the clot and stimulate cell adhesion, proliferation, and clot resorption. Then basophil and mast cell, especially the mast cell, it is derived from bone marrow located near the small vessel. It contains heparin, that is an anticoagulant, histamine that acts as vasodilator and increases vascular permeability. Then there are many proteinases that play their part in the degradation of the matrix. Their release is triggered by the foreign antigen and it plays a key role in angiogenesis and inflammatory process. Then macrophage orchestrate the early inflammation and initiate a regeneration. Epithelial cells of this area where the injury occurred, they release the growth factor and aid in wound coverage. Then mesenchymal cells, they contribute to extracellular matrix and fibrosis. All these activities, they lead to the tissue repair via increased cellularity at the wound site controlled degradation of the damaged tissue, and initiation of the vascular supply restoration, tissue regeneration, or fibrosis. So the major cells that are involved in this activity are leukocyte, polymorph nuclear leukocytes, monocytes and macrophages, dendritic cell, fibroblast, myofibroblast, pericytes and smooth muscle cells, endothelial cells, epidermal cells, and stem cells. The immediate cell to migrate to this area are leukocytes, and they come from the circulating blood. They adhere to the endothelium, migrate into the tissue via focal adhesions, and this activity is guided by chemokines. Within hours, the bone marrow sends the neutrophils and polymorphonuclear leukocytes. They start degradation of the debris or pathogen via the granules and reactive oxygen species, undergo apoptosis, and these are cleared by macrophages that join this area after the neutrophil. And these macrophages may be resident or recruited from bone marrow or spleen, and they serve the activity of phagocytosis of the debris, and they also regulate the healing via cytokines and chemoattractants. Early in the injury time, dendritic cells are also involved in this activity. These dendritic cells may be resident or recruited from the bone marrow or macrophages, and macrophages also. They present the antigen and activate the innate and adaptive immune response. From day 3 to day 4, the mesenchymal cells such as fibroblast, myofibroblast, pericytes and smooth muscle cells that are present in that location or are recruited from bone marrow mesenchymal progenitor, they appear in the area, and they are responsible for fibroplasia, that is synthesis of the connective tissue, tissue remodeling, wound contraction and strength. Then at the same time, after day 3, the endothelial cells from the local venule and bone marrow progenitors, they start to form new capillaries for gaseous exchange, delivery of the nutrient and cell influx. Early in this activity, the surface migration of the epidermal cells or keratinocytes occur from the surrounding epidermis, and they migrate. If the wound is on the skin or injury is on the skin, they migrate across the wound surface. It is delayed by damaged basement membrane, phenotype altered if absent. Then continuously starts from the early time. There is arrival of stem cells, progenitor cells. They come from bone marrow, hair follicle and basal epidermis. They differentiate into epidermal dermal cells, form new vessels or epithelium and regenerate the structure. Here the step 1 of the tissue repair and immune response is described, that is the leukocyte and stem cell migration from marrow or circulation. focusing how immune and progenitor cells, exit blood vessel and enter the tissue. The bone marrow sources are shown in the cross section over here, and here the white blood cells, stem cells, mesenchymal or hematopoietic, they are released from this area to the bloodstream. Endothelial cells form the inner lining of the blood vessel. These endothelial cells, they are forming the inner lining of the blood vessels, and RBCs, they circulate within the lumen. Then a leukocyte exiting the vessel, like here, it is exiting the vessel through the endothelial layer, entering into the tissue, the extracellular matrix. This process involves the activity of integrin or intercellular adhesion molecules on endothelial surface. A rearrangement of the actin cytoskeleton, this one, the yellowish one, this rearrangement facilitates the movement of these cells toward the site of the injury. Interaction between the basement membrane and collagen by using integrin matrix receptor. Here, the migrating cell, they adhere to the extracellular component like collagen, basement membrane, and this anchors the cell and guides it into tissue of immune surveillance and wound healing. So ICAM and integrin facilitate the firm adhesion to the endothelium, and integrin and matrix enable the anchorage to the extracellular matrix. Actin powers the uh, uh, movement through the cytoskeletal rearrangement. This step is essential for immune defense, that is for example, immune defense by the neutrophilic recruitment, tissue generation via stem cell homing, and inflammation and repair. Here in this step of tissue repair, endothelial and endothelial progenitor cell migration is described, that is a vital event in angiogenesis. Angiogenesis, that is the formation of new blood vessels during wound healing. The capillary anatomy over here, it is showing that the endothelial cells, these endothelial cells, they are lining the vessel and they are supported by parasites. These parasites are wrapping around the endothelial cells, and these parasites, they help regulate vessel stability and remodeling. Endothelial cells and endothelial progenitor cells become activated when there is signal from the wound, like angiogenic factor, that are released locally. They migrate out of the vessel and form a new vessel sprout in the direction of the healing tissue. And the key growth factor that is essential for this are vascular endothelial growth factor or VEGF, or fibroblast growth factor FGF, that is released by macrophages and other cells. These factors stimulate endothelial activation, and promote migration and proliferation. Here the assembly of basement membrane is described. This basement membrane is made up of laminin type 4 collagen and perlecan, and it is forming a scaffold around this. Migrating cells move through the extracellular matrix made of collagen 1 and collagen 3. Integrin on the endothelial cell bind to fibrin and fibrinolactin, fibrin and fibrinolactin, and these facilitate adhesion, movement, and matrix remodeling. So this process is essential for vascularization of the wound, tissue regeneration, prevention of ischemia in healing tissue. In this picture, the step 3 of the tissue repair, that is the parasite migration into the stroma. That is an important event in the vascular remodeling and wound healing is described. The capillary, this capillary that is shown in the cross section over here, this one, it is surrounded by a basement membrane, and this basement membrane, just below these basement membrane, there are parasites. There are parasites. These blue cells wrapping around the capillaries. A parasite detaches from the vessel wall and migrate into the stromal space, where it differentiate and contribute to new extracellular matrix formation, including the collagen deposition. The regulatory signals for these are angiopoietin 1 that promote capillary stabilization and parasite attachment, and angiopoietin 2 that helps or induces parasite detachment and migration, and it is essential for angiogenesis and matrix remodeling. Migrating parasites, then they interact with newly forming extracellular matrix component like collagen 1 and collagen 3, contributing to the structural repair of the injured cell. These parasites play a pivotal role in the stability of the vessel, tissue regeneration, and fibrosis regulation. Dysregulation of these can affect healing and contribute to chronic wound or tumor angiogenesis. So this illustration is showing the wound healing in the phase. In this phase, the fibroblasts are migrating to the site of the wound. This is a critical phase in the proliferative stage of the tissue repair. The wound cavity is shown over here. Here, it is containing the fibrin matrix, that is purple meshwork. This purple meshwork is the fibrin matrix that is forming the scaffold for incoming repair cells. Here, a macrophage is present in the wound and it's playing a key signaling role. The signaling molecules are platelet-derived growth factor. It is secreted by the macrophage and platelets both. It chemotactically attracts fibroblasts toward the wound site. Fibroblasts, these fibroblasts that are elongated cells, these blue cells, these fibroblasts, they migrate into the wound Once at the wound, fibroblasts synthesize collagen. This collagen is synthesized by these fibroblasts. They also produce fibronectin, release proteoglycans, secrete growth factor like fibroblast growth factor and transforming growth factor beta. These molecules help rebuild the extracellular matrix and support new tissue formation. The collagen bundles represent newly laid down extracellular matrix scaffold, providing strength and structure to the regenerative tissue. So, to summarize, the macrophage release platelet-derived growth factor that calls in fibroblasts to this area, and these fibroblasts lay down extracellular matrix component like collagen fibronectin, and this set the stage for the granulation tissue formation and eventual reepithelialization. The fibroblasts then differentiate into the myofibroblast, and the signal for this is transforming growth factor beta. They differentiate into smooth muscle actin containing myofibroblast that maintain the bipolar shape and continue migration. So the function or outcome is contraction of the wound and deposition of the extracellular matrix that aid in the tissue remodeling. This detailed diagram is showing the reepithelialization and migrating epithelium. That is a crucial step, and where the epithelial cells migrate, proliferate, and restore the epidermis over the wound. So here the sources of these stem cells are mentioned. The basal layer of the epidermis, hair follicle, bulge region, and sebaceous glands contain the stem cells, and these cells proliferate and contribute to the wound coverage and regeneration. The basement membrane serves as the scaffold for migrating basal cells. When intact, it guides orderly reepithelialization. So, migrating cell move across the wound, like here, the migrating cells are moving across the wound beneath the fibrin clot. This is the scar of fibrin clot. So, they are moving beneath this fibrin clot, and they proliferating cell and replenish and support the migrating front. The dermis below contains collagen matrix, fibroblast, inflammatory cells, capillaries, and this area is called the remodeling matrix. And remodeling matrix area contains some molecular markers. Matrix metalloproteinases, the enzymes that are expressed by migrating and proliferating cell to remodel extracellular matrix and aid in migration. Migrating cells express matrix metalloproteases 1 and matrix metalloproteinases 10, MMP1 and MMP10. The proliferating cells, they express MMP3 and MMP28, while the intact skin show no new MMP expression. Integrin are the adhesion molecules that help epithelial cells anchor and migrate along the matrix and basement membrane. This process is critical in wound healing because the effective reepithelialization prevent infection and restore barrier function of the skin. Delay in this process can lead to chronic wound and scarring. This is triggered by fibroblast growth factor and VEGF. The migrating uh, keratinocytes signal for these migration of the keratinocyte is matrix Q and exposure to the provisional matrix and collagen type 1. The action or process for this activity that the integrin receptor switch to recognize provisional matrix, upregulate metalloproteinases to support migration and matrix remodeling. So the outcome is enhanced epidermal migration, promote efficient wound closure and remodeling. Now we quickly go through the sequential activities that occurs during these cell migration. So first step is the recruitment of leukocyte and circulating stem cells. This trigger for this activity is cytokine activation of the capillary endothelium, and these cell adhere to endothelium, transmigrate through it, penetrate basement membrane and enter the matrix via chemotaxis, and the final outcome is initiation of the inflammatory response with attraction of the repair cells. The polymorphonuclear leukocytes then join, and chemotactic signal is the trigger for this along with the inflammatory cytokines. They rapidly infiltrate the wound, release granules and reactive oxygen species, phagocytose the pathogen and debris, and the outcome is debridement and early immune defense. Monocyte replace the neutrophil after some time, and the trigger for this recruitment of monocyte and macrophages is inflammatory signal and natosis, the dying of the neutrophils. They recruited from the bone marrow and spleen, and they phagocytose the apoptotic cells, release cytokines and various growth factors. Hence, they orchestrate the granulation tissue formation and start of the healing process. Dendritic cells trigger are tissue damage and pathogenic signal, and they present antigen regulate immune and adaptive immunity responses. Their function or outcome is the regulation of the immune system during the repair. Next is the involvement of the endothelial cells. The angiogenic factors are the trigger and signals for the activation of these endothelial cells or migration of these endothelial cells. These angiogenic factors are VEGF and fibroblast growth factor. So the endothelial cells are detached from the basement membrane and sprout into the area of provisional matrix. They form new capillaries that is termed as angiogenesis. Then the parasite basement membrane component involvement, it provides a cue for the vascular remodeling. Its function is to stabilize nascent and existing capillary structure, and the final outcome is to maintain the vascular integrity and support of the angiogenesis that has started. Fibroblast trigger for the fibroblast are platelet-derived growth factor and other growth factors. These fibroblasts become bipolar, migrate through the matrix to the wound, and populate the wound site and secrete extracellular matrix. Fibroblasts then transformed into myofibroblasts, and the action or stimulus is by transforming growth factor beta. So they differentiate, express smooth muscle actin, retain migratory shape, and final outcome is wound contraction, extracellular matrix deposition, and remodeling of the structure. Basal keratinocytes, they migrate through the through the trigger or action or stimulus of the fibroblast growth factor and VEGF. They are released from the basement membrane, migrate under the fibrin eschar between it and granulation tissue. They start the reepithelialization to cover the wound that finally restore the barrier, skin barrier against the pathogens. The migrating keratinocyte signal or trigger is provisional matrix type 1 collagen matrix metalloproteases activation, and it changes integrin receptor profile and produces matrix metalloproteinases. They enhance the matrix remodeling and promote the closure of the epidermis. Stem cell progenitor cells are recruited to this area or migration occur due to injury signals or chemokines. They are recruited from the bone marrow, hair follicle bulge, basal epidermis, and differentiate into the skin or vascular cell type. They are responsible for regeneration of the epidermis, dermis, blood vessel, and skin appendages. Mechanism of migration of these cells is by the chemoattractants, both the endogenous or exogenous. Some exogenous are bacterial products, while the endogenous are cytokines, chemokines such as interleukin 8, complement system activation that will lead to formation of C5a and arachidonic acid metabolite, especially leukotriene B4. They bind with these chemoattractant bind with the G protein coupled receptors, and there is activation of second messenger system. GTPase of the RAC Rho and CDC42 family are activated, and that will further activate the kinases, and there is, due to this kinase activity, there is polymerization of the actin, and polymerization of the actin at the leading edge of the cell and localization of the myosin filament at the back. So they start moving by philopodia, or the back area, that is lamellipodia. The myosin condensation is termed as lamellipodia. So here they start toward the migration toward the site of the injury. Later, the remaining part of the cell, it also moves along with this front edge or front leading area like a car, that is front wheel driven car. The leading edge of the moving cell, it adheres to the extracellular membrane adhesion receptor that are integrin, and the focal contact anchors actin stress fibers against which myosin pull to extend or contract the cell body. And a Rho family of glycine triphosphate GTPases, Rho, RAC or CDC42, they form an adhesion plaque. And finally, older adhesion at the rear end are weakened or destabilized. allowing the trailing edge to retract and a new adhesion plaques are formed. So, like this, the cell migrate and it moves on toward the chemotactic stimulus. So coming back to the sequential cell activities in skin wound healing, the migrating cell, any of the migrating cell, especially the keratinocyte or fibroblast, there is extracellular matrix interaction and there is activation of the integrin. Leading edge of the cell adheres to the extracellular matrix via integrin, form a focal contact plaque at the front and focal adhesion beneath the cell body, and this enable traction and forward movement of the cell through the wound matrix. The focal adhesion formation or cytoskeleton triggers the actin-myosin machinery, and actin is stress fiber anchored to integrin. Myosin pull against these to extend the cell body and propel cell forward and allow retraction of the trailing edge. Intracellular signaling protein are triggered by integrin cytoplasmic domain signaling. They regulate adhesion, actin cytoskeletal organization, and matrix interaction, and coordinate the cell migration, matrix remodeling, and directional movement toward the chemotactic stimulus. Epithelial, endothelial, or mesenchymal cells are triggered by integrin extracellular matrix binding. Cytokines, growth factors such as FGF and VEGF, and transforming growth factor beta. The integrin transmit intracellular signals that regulate survival, proliferation, and differentiation via cytoskeleton and the signaling cascade. Then there is the outcome that maintain the cell viability, promote specific functional phenotype in the healing tissue. Integrin associated signaling machinery is the stimulus for this is inside out signaling via DDRs, tetraspanins, growth factor receptor, and their action is to modify integrin affinity for matrix component and influence cell response to extracellular matrix. The final outcome is fine-tuned adhesion strength, migration capacity, and remodeling of the matrix. All cell types involved in wound healing, trigger for these can be integrin growth factor receptor crosstalk, and these are activated by shared signaling pathway that uniquely organizes cytoskeleton via integrin. They coordinate tissue structure, drive cell shape changes, and control the differentiation. Epithelial cells may undergo epithelial to mesenchymal transition, and the signal for this is changes in cell to cell and matrix adhesion or mechanical stress. The action is cytoskeletal reorganization that lead to epithelial to mesenchymal transition, and it enables reepithelialization, migration, and eventual restoration of the skin architecture. Here, the specific type of the cell, their time of arrival into the wound, origin, and function in the healing process are summarized. Neutrophil reach the polymorphonucleoside reach within hours from the bone marrow, and they start to clear the debris and pathogen via degranulation, and reactive oxygen species undergo netosis and cleared by macrophages. Then neutrophils are vanished, and monocyte and macrophages, they come from bone marrow and spleen and start phagocytosing the debris, secrete cytokines and chemoattractants, orchestrate the granulation tissue formation and healing of that area. Dendritic cells that are resident to that area, and they come in early time. Their origin is the skin and bone marrow, and they are responsible for presentation of antigen, modulate the immune response. Fibroblast and myofibroblast start to appear at day 3, 4 and onwards. They may be present from the local population or recruited from the bone marrow. They are responsible for remodeling, contraction and tensile strength to the wound. From day 3 onward, endothelial cells also, under the various influences, come from progenitors, or some local also sprout to form new capillaries, that is angiogenesis, and it support the nutrient and gaseous exchange. Keratinocytes are the early cell for the migratory process. They come from the skin, hair follicle and basal layer stem cell reserves, and they are responsible for reepithelialization by migrating under the fibrin scar and over the granulation tissue. Stem cell progenitor cells, their contribution is ongoing. They come from bone marrow, skin follicles, and they differentiate into fibroblast, keratinocyte, endothelium, and all this contribute to regeneration of the tissue. Thank you for watching this video. If you have any query, please write in the comment box.